Hello everyone, I hope you all would be fine. From today, we will be mixing the various topics from different chapters together so that we can expedite the completion of this series of important topics. So we will be taking important topics of different units together. Today, our topic is from this unit 2 of animal physiology, and this topic is absorption of digested food. This is an important topic and also a complex one. Other topics you can find easily in the books, but this is a bit tricky one. First of all, let's see, where does this absorption happen? It happens right from the mouth till the large intestine, but most of it, about 90% of it, is happening in the small intestine, and the rest 10% is collectively happening in the mouth, stomach, and then the large intestine itself. Now let's take a look at these aspects. In mouth, few drugs get absorbed. In stomach, there are various things like ethyl alcohol is there, aspirin means it is a kind of drug, then glucose up to a certain level, this gets absorbed. Likewise, in the large intestine, various drugs, vitamin K is important here because vitamin K and vitamin B complex, these are produced by various bacteria in the large intestine and they are being absorbed through the large intestine itself. Then water and drugs, various drugs, they are absorbed through the large intestine. So the major chunk of it, which is small intestine, that we are going to discuss. But let's first discuss the various modifications which are there in the small intestine. First is that its length is approximately 6 meters. It means it is providing much better surface area for absorption. Then it is having various folds which obviously increases the surface area. Then there are various villi. This is the villi, this is the structure of villi. These are small, 1 to 2 millimeter structures which are rod like structures, and these are made up of the enterocyte cells. These are having these finger like projections. These finger like projections are microvilli. We will discuss that. Besides this, the most important thing here is that we are seeing this capillary network, and then in between there is this lymphatic capillary which is known as lacteal. We will see the roles of these, because the things which get absorbed from here through this villus, it directly enters the blood or the lymphatic system, and this is the major thing, the villi. Then we are going to study the microvilli. These are also the modifications. As we can see on a villus, there are various microvilli, these finger like projections on the enterocytes. These are in turn increasing the surface area, and this is a very logical thing. Now let's move towards the actual mechanism of different things like carbohydrates, proteins and lipids which get absorbed. First they are getting digested, and then these things get absorbed through these villi and microvilli. Now let's first take the carbohydrates. We must be knowing this thing first. This is the intestinal region. This is the enterocyte here. This is the enterocyte, and it is having these projections known as microvilli. Its magnified version is this. This is the magnified version. It is having these finger like projections, and then this whole cell. It looks quite similar to this. This is the same thing at micro level. Now let's see what's happening. This is the enterocyte. This is the intestinal lumen side. This is the basolateral side, where we can see the blood capillaries and lacteal. This side, this internal side, this is here, this part. Now let's first see what is happening with the carbohydrates. With respect to carbohydrates, we are majorly having the the digested part is majorly having galactose, glucose and fructose. First, let's see galactose and glucose. These are getting transported through active transport, but this kind of active transport is known as secondary active transport. Now let's break this term up. Why is it known as secondary active transport and how is it active? Just keep a focus on these things, these concentrations. In the intestinal lumen, glucose is less and sodium is more with respect to this. This being less or more is with respect to the enterocyte, means inside cytoplasm of the enterocyte. Here glucose is more and sodium is less. Now what is active transport? If we see, it is the transport from lower concentration to higher concentration of something by using energy. Let's see. Here what is happening, glucose here is less, it is going from lesser to higher side, right? So it is an active transport. But where is the energy? Energy is not being used. No ATP is here which is getting utilized. That's why it is known as secondary active. We will go into it a bit more in a while. But first let's look at it, that it is a co-transport kind of thing. Here there is a protein which is transporting both sodium and glucose. While sodium is going from its higher to lower concentration across the gradient, with the same thing, by using the same energy, glucose is transporting from lower to higher concentration gradient. Now why is it known as active transport and where is the energy? One aspect we studied that it is from lower to higher concentration, but where is the energy? Where is the use of energy? The use of energy is here. We can find that it is in the enterocyte we are having low concentration gradient of sodium. But why is it low? Because here these sodium ions are getting pumped out of the cell and potassium ions are getting in, and here it is the proper active transport. So the energy which is being used here is actually being used to pump these things out and make this concentration gradient low. That's why from here we can find that these sodium are coming inside the enterocyte. Means that's why it is secondary active transport. It is active because with respect to glucose it is going from lower to higher concentration gradient, but it is secondary because the energy utilized at some other place here for creating a concentration gradient here is being used for this transport as well. That is why it is known as secondary. Secondary means energy is not directly being used, but it is being used somewhere else, and that effect is being shown. Means here it is being used somewhere else and that effect is being shown here, and that effect is that of lower concentration gradient of sodium inside the cell, and that is being used for pushing this here, and by this the glucose is also getting into the cell from lower to higher concentration gradient. That's why it is known as secondary active transport. From here glucose moves through the diffusion, and that is a facilitated diffusion through this GLUT protein. Facilitated means there is no use of this energy, but some protein is being used that is facilitating this. That's why the name is facilitated diffusion. Now this must be clear. Now let's move to another part. Fructose is always going through facilitated diffusion via this protein from higher to lower concentration gradient. Diffusion is always from higher to lower concentration gradient. and it is a passive mode of transport, but when it is facilitated, it means some protein is being used. These permeases are being used, and from here means from fructose, from this enterocyte to these blood capillaries, here it is getting through simple diffusion. Now let's come to proteins. Proteins are digested into amino acids, and amino acids travel through secondary active transport here with sodium or calcium into inside the cell. Then from inside the cell, by simple diffusion, it moves to blood capillaries. And from blood, what happens for all these things means the carbohydrates and amino acids, carbohydrates including fructose, galactose, glucose, they are going through hepatic portal vein to liver, and from liver they are going to blood. Means here they are moving to blood in the blood capillaries, but not in the major circulation. First they move to liver, from liver they are moving to the blood circulation as per the NEET. There are certain mechanisms for that also, but for that much of detail we must not be entering at this point of time. Now let's go for lipids. Lipids are actually heterogeneous group, means they are made up of, say, fatty acids, and they are attached with a glycerol molecule, which is actually an alcohol molecule, and they are insoluble in water. So they are heterogeneous group, and simply speaking, they are consisting of fatty acid chains attached with a glycerol molecule. In a very simple way we can say this, or we can say this: the end product is either a glycerol molecule or fatty acid chains, or diglycerol, this one fatty acid chain and glycerol molecule, or this is monoglycerol or diglycerol, or we can say diglycerides, means two fatty acid chains and one glycerol molecule. And fatty acid chains can be short chain fatty acids, which are actually dissolvable in water up to a large extent. That's why by simple diffusion it gets into the enterocyte, and from there it gets into the blood capillaries. But with respect to long chain fatty acids or monoglycerides or diglycerides, say monoglyceride means glycerol molecule and one fatty acid chain. Diglyceride means two fatty acids and one glycerol molecule. What happens? They are large molecules and they cannot directly diffuse. So what happens? First a micelle is created with the help of bile salts that helps in easy pinocytosis into the cell. From there bile salts get separated and they move to liver, and this circulation is known as enterohepatic circulation. Then these long chain fatty acids, monoglyceride, diglyceride, they get converted into triglycerides in this smooth endoplasmic reticulum. Then that is packed into a chylomicron made up of phospholipids, cholesterol and lipoproteins. That goes to lacteal, which is here, this vessel, and from this lacteal it moves to thoracic lymphatic vessel, then to blood plasma, and in blood plasma there are various enzymes which break down this thing again into fatty acid and glycerol, which is being used. So this was the overall concept of absorption of the various digested food material into the intestinal lumen. From here things get assimilated and reach to their destined cells. This was all about it, and let's hope that you liked the concept. Do join our Telegram channel and also do subscribe our YouTube channel. Thank you.